Good afternoon, everyone. Welcome to the Wake Forest Center for Artificial Intelligence Research Artificial Intelligence in Action seminar series. I am Metin Gürkan. I'm the director of CARE, and it's a pleasure to introduce today's distinguished speakers, Dr. Javed Mostafa. And then Dr. Mostafa is professor and dean of the Faculty of Information at the University of Toronto. His research spans multimedia information retrieval, personalization, user modeling, and cyber infrastructure for learning and research. Prior to joining the University of Toronto, he served as a professor of UNC Chapel Hill, where he founded the Carolina Health Informatics program. And many people know this program as the CHIP program. It's a remarkably successful and well-known program. And then he has also held leadership roles at Indiana University and has published extensively across the fields of information science and technology. Dr. Mostafa will present on the role of artificial intelligence in transforming biomedicine and healthcare from hypothesis generation and clinical interventions to patient engagement and research evidence production. He'll highlight both the opportunities and challenges of deploying AI across this continuum and conclude with insights on building and sustaining learning health systems, which is very famous here at Wake Forest. Feel free to enter your questions in the chat if you are joining us online, and then we will address them following the talk. And then we have also several people in the room, and then they will be also asking questions. With that, let's warmly welcome Dr. Mostafa.
Thank you so kind.
I look forward to your talk.
Thank you. Really appreciate it. Thank you so much. So yeah, let's try to kind of keep it a bit informal. I will try to pace through today's talk and give space, a room for discussion, and if you want to get a bit more elaboration, I'd be happy to provide. So I'm so grateful to my friend Metin and honored to have the chance to come to provide this presentation. And I have been working in this area of biomedical informatics and health informatics for several years. As you can see, I'm an old man and getting older by the day. I am currently involved in more of the secondary analysis and research informatics side of things, but some of the projects and the techniques and methods I'll be discussing are also very timely, I think. Really, I come with the spirit of, I mentioned this to Metin, to explore collaboration and create new projects, hopefully. Metin has been a wonderful collaborator and a role model for me in some ways, just to watch him create initiatives and being so professional in terms of managing big, complex initiatives. So I could not resist the chance when he said, come please and provide us some talk and meet some people. So I'm going to structure the talk as kind of a AI ML applications in medicine and biomedical problems and challenges. I know biomedical informatics may seem like kind of a relatively small discipline, and there aren't many universities that have departments in biomedical informatics, as you know. But my grounding, really comes from more of a applied computing sort of type of work. My background is in computing, but and computer science. But when I was early in my career in Indiana University, as you may know, Indiana University has a very famous center called Regan Street Institute. It's very well known for in the biomedical informatics area for developing standards and data protocols, pioneering some of those methods and techniques for electronic health record systems and electronic data in medicine. So I got started after my PhD in Indiana University, and I started working closely with people in biomedical informatics, and that's how I got exposed to the area. I never thought that I would spend 20 plus years in that field. And when I came to UNC after IU, I had the chance to create this center, CHIP, Carolina Health Informatics Program. And then that blossomed into a degree granting and training program and developing connection with Duke and Wake. And it was a lot of fun. So the types of projects I'll be discussing, they're not necessarily advancing fundamentally, probably computing and AI methods, but they are trying to demonstrate how AI and advanced computing methods can help in the whole pipeline of biomedicine to all the way to translation and application. And so I'm going to start with a mapping of AI ML to biomedical health informatics. I will demonstrate some research projects and problems. I won't go too deeply into the methods, but I will show the papers related to. Most of the things I will discuss have been published by me and my collaborators, and I will point to those papers, and then I will begin to wrap up thinking about where do we go from here, what's the next level of advance in application and advancing ML in biomedicine, and then also conclude with some sustainability discussion in terms of how do you scale this, how do you manage these kinds of research initiatives. So I don't know, how many of you had a course in AI? Some of you had, right? So AI is actually, nowadays, it's the hottest thing, but of course, it's a very old field, right? And it's depending on how you count, it's at least probably 70, 75-year-old field. But when you look at the early days of AI, and AI is bigger than machine learning, right? So sometimes we confound the two things. AI has many other things besides. Anything that makes systems smart is AI. That's a big umbrella. And under AI, ML is one big area, but any tools, any technologies, even hardware innovation that make the technology smart is trying to advance AI. So AI is much bigger than ML. But when you look at the founding early days of AI, like rule-based systems and knowledge-based systems and expert systems, early days of innovation, the use cases for AI, just to demonstrate those new methods, new knowledge representation, new AI systems, were healthcare. Okay. So we think of AI application today, oh, it would be nice to apply AI in healthcare. But AI and healthcare develop together, the innovations, right? And so earliest domains were healthcare. You can obviously, nowadays, by the way, maybe some of you, as I am talking, you're talking to Uncle Google and you're checking, that's great because you can validate everything I'm saying. Of course, I also talk with my uncle, so just to make sure we're all aligned. So One of the most famous use case was Mycin, which was doing prediction on infection, bacterial infection, and doing that automatically. This is a very early days of AI, so Shortlift, Edward Shortlift was at Stanford. Those people were working on. Also, notice my comment there. Even in these earliest days, the concern was it's not just prediction, you have to explain the prediction. So the backward chaining logic was very appropriate for that because you could actually then show the path of reason and logic. You start with prediction and then you work back to find all of the evidence that predicted the infection. And this was done for bacterial infection, Mycin. Mycin is like a very famous example in AI. It's used in many AI textbooks. If it's comprehensive, you will see. So obviously they try to generalize Mycin to not just a bacterial infection, to many other case problems, where you could then do prediction using knowledge-based approach or rule-based representation approach, and that was called eMycin. So, but this is how old AI is. So AI was termed by John McCarthy. Some people think it's Minsky, but it's actually McCarthy. Those two guys are considered the fathers of AI, Marvin Minsky and John McCarthy. John McCarthy actually had a workshop or a little conference where he coined the term AI, 1955. Okay, and these systems that I just finished talking about are 72, 74, but then another very famous use case which has become very important today is a chatbot that was invented in 1966 called ELIZA. And the use case there was also psychiatric analysis. That was also in healthcare. So these are very important foundations for AI, and there's no wonder AI is so important in healthcare. But note, please do note the fact that you guys are the young next generation. The impact that we expected 70 plus years ago, we still don't have that impact, the outcome. Right? There's a lot of promise, but at scale, the impact is still not there in terms of seeing the results. Only recently, there have been a lot of talk also. You saw the interview Gates gave, Bill Gates. He said that there won't be any doctors needed in about 10 years or so. No teachers needed in about 10 years or so. Very famous quotations and statements are given, but let's see where it goes. All right. So now let's take a pause. I want to go a bit faster, of course. Let's take a pause and think about where AI ML can be practically helpful. And let's think about the biomedicine and healthcare together, because healthcare is very, very dependent on biomedicine, meaning that healthcare is an evidence-anchored field. Doctors have to get relicensed on a regular basis, at least in the US, because the evidence, the way you practice, how you treat your patient, what drugs you prescribe, continuously changes. Okay? And how does it change? Based on research. And where does the research come from? Biomedicine, right? So this is why I love this field broadly, because research, the distance between research and practice, even in the highest level, is very short, right? Doctors are supposed to know the most important findings in terms of how to treat a patient, supposed to, not always, by keeping up with their field, right? By reading the evidence in JAMA, for instance, right? So let's take from discovery, like where the evidence is found, the research. When I say discovery, think research, okay? Let's go from discovery, diagnosis, then to treatments and interventions, then to actually delivery of the care, engaging the patient, think about the outcomes, of course, then think about what happens after you do the delivery to the patient. You've treated the patient, you've seen the patient, you've diagnosed the patient. These days, what happens after that? You get a lot of data, right? Theoretically, at least, you should have access, all of you. I don't want to cause trouble, but yeah, as informaticians, you are supposed to get access to that data for good reason. Why? Because you need to do secondary analysis. You need that data for your research. Right? We are closing the loop already, right? We are started with research, going to end with secondary analysis. We're improving the treatment and the comparative effectiveness, the quality. So where does AI ML fit? AI starts even at the discovery stage. I will show some example why it's important in the early research stage, sometimes called hypothesis generation, right? Assays, you know what assays are? Assays are biomedical methods, experimental methods that are standards that have been tested, the protocols for doing experiments. They are very important to biomedical researchers. Those that are trained as biomedical PhD, MD PhD, they know their assays, how to run these experiments. And these assays are published in research papers. These are the methods, right? Mm-hmm. Increasingly, even scientists do a video of how they run the assays, the experiment, the video, they publish the video. So you can follow all of the steps they have taken. This is the essence of science because you have to replicate their research, right? So even in the assay, you can apply AI. Why? Because if you apply AI, then you can repeat that process. Okay? The assay automation is becoming a very important area of innovation. There is a center at Toronto called the Acceleration Consortium. Acceleration of what? Acceleration of all of these complex workflows, including assays. Right? So watch this space of assay automation because this is a very hot area. It's a very important area. Agentic workflows. So assay automation, workflow automation need distributed AI because the machines, from one step to another step to another step, the machines are separated sometimes, even by distance. They're not close to each other. You take data from one place, you take it to another place, you run it in another instrument. All of these different places need to be intelligent. Guess what? You have to use then multi-agent distributed AI communication between them to maintain that synchronization and calibration. Okay. So by the way, another example where we're so excited about, I sound like an old man, I am an old man, agentic AI, right? We're so excited. But ask your favorite uncle, Google, how old is the field of distributed AI? That's actually agentic AI. It's actually, at least a few decades, at least 3-4 decades old already. Right? So let's not reinvent stuff. I mean, we can say I'm doing agentic AI and you look cool. But the basic underlying area has existed for a long time. There have been people, very famous people, have done a lot of work on multi-agent frameworks and how does that work and all of the challenges, right? Treatment, intervention, lots of AI, ML applications I will show, and challenges and applications. I will show some examples. We all know about risk prediction, risk analysis, precision medicine.
where AI can be very precision medicine for me, the way I understand it. Again, my memory is failing, so I need one or two good examples. Is when you walk in today in a complex medical center, like a cancer center, and you have prospectively a cancer, they can bespoke design the treatment for you, not treatment for a population. To me, that's a gold standard precision medicine. And that's, by the way, I wouldn't say routine, but it's happening on a regular basis. Routine in Wake, for sure. Routine in UNC, for sure, but probably not in rural areas. So if you have breast cancer of a certain type, they will genotype you, find your profile, and create a treatment just for you. That's where medicine will go eventually, right? And think about it. That's a perfect domain for AI ML application, right? So we have done some work with Lineberger Cancer Center. I will show an example. So how do we then manage the treatment pathway? Because treatment is not just once, right? As you know. So treatment adjustment, treatment management is a complex problem. How do we deal with that, right? Then we have, once we've designed the treatment, adjusted treatments, we have delivered treatment, guess what happens? Patients need to be tracked. Patients' outcome needs to be tracked. I don't know, how many of you know the area of PRO? Again, if you're looking for a new area to jump in, it's a fantastic area. Patient-reported outcomes. Why? Because most patients are not in the hospital, not inside a facility where you can track them. Most patients are outside, walking about, But doctors have done their treatment, delivered the treatment to them. They have no idea if they are having efficacy. How can we improve that? That's PRO. So remote patient tracking, patient-reported outcomes, there's a big center. Also, I'm sure you guys have someone working on that area, I'm very sure. So this is a very important area. And beyond that, patients also need to be trained to manage if they have complex comorbidities, complex chronic combination of diseases. It's hard for patients to know how to manage their treatment, right, themselves. So patient education, as you know, I don't know if you have a patient education center, pretty much all places, large medical centers have patient education. It's a hugely important area, but it's ripe, very ripe, just like education broadly is ripe for intervention for AI ML. Patient education is very, very low-hanging fruit, believe me. And it's not taken seriously enough, in my view, but it needs to be. And then comes the whole big area of you have dealt with diagnosis, you've dealt with treatment planning, you've delivered the treatment, you want to track the patient. Now you have a lot of data that got generated in the process. So primary use of data is when you have a doctor use the data to diagnose, treat, and manage the patient. Secondary use is when you take that same data and you want to do research on that data, and you want to do quality improvement with that data. That's called secondary analysis. So there's a whole bunch of things. Some people have made, in our view, by the way, Mateen, we were talking about Peter Emby.
Yeah.
Peter Emby. There's a guy who just walked about here, as I understand, has spent his time here. He's considered one of the founders of CRO, Clinical Research Informatics. That's how he became big, in my view. What is clinical research informatics? is doing informatics to accelerate and do research in innovative ways. Right? See, that's research? Yes, because the data and infrastructure are so complicated in medicine. I don't have to tell you. We need innovation, how to, I'll show some examples, how you get access to the data, how do you create pipelines on the data, how do you do, feature engineering to modeling to testing. In a way, it's kind of like the assay automation problem. How do you do that in smart ways? That's CRO, right? And then there is also established research areas like comparative effectiveness research, right? Some people you'll hear sometimes from clinicians, they don't get very excited about QI or Q&A projects because they poo-poo it. They say, yeah, it's not really research, they're just doing some fragmentary things here and there. These are for kids, like, sorry, guys, for assistant professors, and it's a QI project. But the big daddy of QI is comparative effectiveness research, CER. Very, very important, right? What is that? That's basically you're taking a disease and you're taking that disease and you're asking questions like that same disease as treated using guidelines and standards, which is supposed to be normalizing the process, right? Standardizing the process. At UNC, a CVD problem, let's say, cardiovascular disease, at UNC, you can use the diagnostic codes for a certain type of CVD and see what's the outcome longitudinally for a sample population at UNC and compare that with Duke and wait. Not just that, I'm simplifying. You want to then compare what was the cost. And if there's a big cost difference, you ask the question, what's happening? Why is the delta so large? It's the same disease, it's being treated using standards and guidelines that are well known for that disease, but the cost differential is too large. That's a classic CER problem. And then you start asking questions like, are the medications different? Some people have made their whole career just asking questions about medication cost inside CER, right? And have become famous, And so this is an important cost for treatment medication. Obviously, it's an important problem. Like a cheaper drug, if it achieves the same thing as more expensive, of course it's going to be an important finding, right? Anyway, so let me go through these quickly. Discovery. This is, to me, it's a very important problem, close to my heart. That's why I actually took the time to write something on the board. So that's a hypothesis. And you want to discover associations and hypotheses. Sorry, I didn't want to make a lecture out of today's presentation. But
So we are enjoying it. Yeah.
Okay, so what is this A, B, BC, AC? What the heck is that? Well, bottom line is biomedicine is very complex and you're always looking for very close associations between phenomenon or key entities in medicine. Key entities meaning like association between genes and expressions and phenotypes, and association between genes and functions, associations between genes and diseases. These are important things in biomedicine that you can just swap the entities with the association between a molecule or a drug and disease. You're looking for entity-to-entity maps and associations, always, as a very core problem. So you find an implicit association between an entity A and B, okay, by looking at certain evidence. On this side, you find another implicit association between B and C in another literature, right? You say, maybe there's a relationship between A and C, right? Did anyone do that as research?
Right.
So some people became very famous doing it, right? And this person founded the field. What is it? Let me find his name. Swanson. So if you love hypothesis discovery, this is the classic paper. So he discovered that there is so Raynaud's syndrome, Raynaud's disease is a very complex disease, right? And scientists were looking for what's the intervention? How do we attack this disease, you know? And so he found a lot of paper that talked about A and B that connected B being fish oil. Then he read a lot of paper that connected B and C being the intervention. Okay? He said, but no one is talking about fish oil having any kind of effect for managing Raynaud's disease. There was no empirical scientific evidence. Right? He did it manually, Swanson. Right? He said, what if we build a software that finds A to B, B to C, and then says, you might want to look at A to C. And guess what happened? They actually later did scientific experiments with fish oil and Raynaud's disease, and they saw that it actually helps manage it.
Wow.
Boom. Right? So why is that important? Let's go back. That's important for a very hypothesis discovery like that is really important. The reason is, do you see this number?
Per minute?
Yes, yes, sir.
So that is the new goal for you.
Maybe with Uncle Google's help, I don't know. This is the world's largest biomedical database. And we're so lucky, even today, I checked this morning, they didn't shut off PubMed yet.
No.
Okay? PubMed is still on for business. Okay? But look at the curve. Okay? PubMed grows at an exponential rate, right? That's not the most interesting thing. The most interesting thing is the red line. This was published in Nature, one of the Nature journals. It basically says the number of new papers that were published each year continues to increase, especially in specialized topics like cell cycle, for which it is, listen carefully, no longer possible to read all the new papers that were published. Think about it. You're a scientist in cell cycle. And you say, I'm going to do it like my advisor did, you know, find all the good papers, read them carefully, and then I'll be ready to innovate. You can't.
No.
You can't do that anymore. And we've known that actually for a decade now. A decade, right? We don't admit it because it's embarrassing to admit it. Right? This was published many years ago, by the way, the curve, the cell cycle curve, right? So what's the relationship between this and Swanson? What Swanson pointed out that the evidence is there, we need analytics to discover the map, the association. Not necessarily we don't want to read them, but read the papers that are important for that hypothesis. It's a different way of engaging. And what did I say about healthcare? It's evidence anchored. And if we can't have enough time to read the evidence, again, it's kind of embarrassing. Not a lot of people will admit that I actually don't know the evidence because I have a life. I can't. I mean, if I read papers all day, then I won't be able to. Right? Now comes the interesting part. There is a whole field that has appeared early, when Swanson published, called text mining, literature mining, usually called literature mining, but also nowadays called text mining. And you think that this is not a serious field? This is a very serious field. It is. I think I will end my talk to show what's happening today in literature mining. Right? Well, the challenges are many, right? Because the way we publish evidence, the way we express our science is actually very complicated for us to, even after we find papers, to absorb and understand and create new hypotheses from it, right? So take the most important dogma in biomedicine, which is how does genes, DNA, control, influence the proteins and protein pathways? In other words, most of our important functions in our body, right? And that central dogma is all about finding associations between entities, right? Gene names, the functions, the phenotypes, and all of the phenotypes controlling a particular expression. This type of association discovery, as represented in science today, is hard for us to absorb. And not just because of volume and scale of the evidence, because they're presented inconsistently. Okay? Protein names, if you just take protein names, right? The scientists describe them in very, eccentric ways. There's a protein called fruity. It was called fruity for a while, but now it's called fruitless. Really, this is a scientific name for Drosophila to control sexual identity. It was discovered many years ago, but it keeps evolving. The name of the protein keeps changing, right? So for us, even on a machine-oriented way to track these kinds of evidence becomes very complicated because of the way we present and express our science. And so there's a lot of work to be done in text mining to manage these kinds of complexities. And you've heard of some of the methodologies that we now apply for unstructured text in EHR. We talked about named entity recognitions and using classification mappings and network analysis and things like that. They are important in a bigger field of scientific evidence generation and managing that complexity, right? So let me go a bit faster. So we published papers a long time ago. Actually, this one was done with the Bayes network, using essentially some established ontologies and vocabularies to close this gap between genes in case of rare diseases and diseases, finding these kinds of associations automatically using inference networks, right? And so as part of that, we also had to improve our NER. So these are papers we published. So this one is probabilistic model for protein name detection. And because I just gave examples, protein names are not easy to deterministically identify because the same protein is called different in different places, right? So you have to look at the embeddings. You have to look at what LLM looks for, like the context of a long text. And you have to look for, once you find the entities, you need to create the probabilistic maps, right? Every edge is a probability of relationship associations based on the evidence using the summations that I'm showing here to then do these association trees and then have a gold standard to see if the association you found is correct using these kinds of networks, right? All right, so that's one, and I'll go a bit faster. For discovery, So I kind of did it the other way, which is, imagine you found a corpus and you want to do a hypothesis discovery. You can apply networks like inference networks, like the ones that I showed. Before you even read, you find the hypothesis, right? But here, what I'm showing is, how do you find the corpus, right? To target, right? Which corpus to target, right? So, you know, PubMed has, I don't know, near 40 million. And remember, 2 papers per minute. So there is no way I can find even this problem, by the way, is not an access problem. It's in biomedical informatics, it's called a findability problem. Before even you access what and manipulate data, you need to know what data to manipulate and access. So these clusters are done using something called unsupervised scatter-gather. So that's an algorithm that was developed by statisticians a long time ago. But we apply scatter-gather in this context because it has to be, you know, probably a bit about machine learning. It can't be supervised learning because I don't have any training set. If I had training set, I would know which cohort to go and look for, right? So this is unsupervised. You have to find these clusters without training, right? So this is unsupervised scatter-gather. But what's cool about this and really challenging, the scientific challenge is, how do you do dynamic unsupervised clustering, like user-driven, user-directed unsupervised clustering? That's where scatter-gather comes into play, because just the high-level clusters is not enough. So this is cell signals, proteins, you know, and this is genetic, population, genome. What I want to do is not look at these clusters anymore. I want to look at these two only. I want to combine them, in fact, and then find smaller clusters in them, right? So I want to basically do it dynamically. Right? This is a problem, by the way, I present to my students. If Google, Uncle Google, who I admire a lot, is so powerful, how come they don't use the most powerful sense for humans we have, which is a sense of seeing as a perception? Google isn't visual. But it's the same thing. You may ask, why do we use diagnostic codes that are not very descriptive for diseases? I don't want to be too cynical about it, but you get my point. Google is not actually, at the end of the day, I know we are being recorded, not necessarily geared toward giving you the best scientific answers. Google is there to make it easy for you to find a restaurant or find the best price for something, you know. So this is another warning that we don't want to use these kinds of tools and databases to do serious research, right? So, but there is room to do research informatics. This is research informatics, by the way, to improve the way you help the researcher to hone in on the right cluster of documents, because the document space exponentially grows, and then to apply hypothesis discovery on that to be able to then conduct their scientific work, right? Okay, that is the paper. This guy finished a PhD 3 years ago, Michael, and I'm proud to say he founded a company. So I was a co-founder of the company called Semantics. So that does these kinds of visual analytics, user-driven analytics. And you know, if you catch me after the talk, I will say why I left the company afterwards. So anyway, the company is doing well, but I had to leave it. So anyway, diagnosis is part of that whole discovery cluster, because we want to do an analytics on large, complex data sets, right? One of these is close to my friend Mateen's heart. I wish I had found Mateen when I was doing this work, but now that I found him, maybe I pick it up again. One of the classic problems in imaging sciences, at least in biomedical informatics, is how do you analyze complex imaging data in a way that makes it easy for you to identify early stage or do even early phase or do even staging of diseases. How do you classify and stratify people in terms of disease states, right? And so we've developed systems published, and this is a place that I think my team calls home, Mackay. How do you do classification for, this is a very classic problem, right? You have normal clusters, you have MCIs, and you have ADs. How do you separate them, right? And so we used a generalized version of support vector machines because support vector machines are binary classifiers. Obviously, we have 3 classes. To do this in a very efficient, effective way, this has been superseded. We got about 8.9 F-score. There have been other people that have come, and now we have 0.95 F-scores and things like that. Anyway, the idea is this admin data set, by the way, if anyone is in imaging science and want to do research, I would be happy to collaborate with you because this is one of the largest image data set for Alzheimer's and dementia and extremely well managed. And you have multimodality with data, omics data, alongside the brain image, MR data, and you have longitudinal data. It's amazing. So, but the techniques that I've described here are relatively straightforward, but the purpose is important because you want to improve the stratification. And so even 0.95, guys, by the way, is not good enough. You really need to push further, right? So other examples, of diagnosis problems, this is an interesting problem because we not only need accuracy, we need scale and we need efficiency. When, again, the problem is data set is very large, especially in imaging, and how can we gain scale, right? And this is an example of using different ML, so unsupervised, supervised, and then distributed agent-based approaches. And we had published, let's see, paper is, what is the paper? I didn't put the paper here, but you can search. This is the important finding on scale, because you're dealing with not one agent, not 100 agents, we're dealing with 100,000 agents collaborating, right? To gain that scale, parallelize the process of classification, right? And we want to hone in on diagnosis as fast as possible on a very large data set, right? And we're showing some interesting phenomena when you do this in a large scale. What happens with multi-agent behavior. So ACM transactions on information systems, I believe is the paper on that one. So anyway, treatment and intervention, let me go a bit faster. How much more time I have? 10 minutes? 5 minutes? Okay. So this is another project we have done on treatment adjustment, treatment planning. Same with the idea of you have patient stratification and patient clustering. This is a system that Duke used to have on psychiatric care called MindLink. And MindLink in psychiatric care, if you're really interested in linking outcome to treatment, psychiatric care is very interesting because they rigorously collect outcome data and they link the, literally, if you walk into a psychiatric clinic, usually they will run a test on you and collect the data and store it just to track how well you're doing. And this is what happens when you plot how well you're doing on combination of treatments, drugs you're taking, and they plan using something called a CGI score. That's the outcome, right? Well, what we did interesting in this treatment planning is we can predict based on this previous sets of data, if I give you a certain combination of drug, if I change your treatment, how well are you going to be doing in the future? Imagine the doctor having that kind of control to change the treatment, not just on your record, by the way, the cohort that you belong to, the patient population that have similar combination of drugs, right? And so that was published a while back, and so I'm showing that result. And on we go. This is one of the more recent work we did on AI explanation. We built a framework for morbidity. This comes from AMI project.
Right.
Yeah. So this is a framework for explanation. We think explanation need different dimensions, XAI, such as doctor needs to understand what the prediction is. Doctor needs to trust the system. Doctor also needs to make use of the prediction and be able to utilize it in clinical care. And we built a system which produces, using Shapley curves, explanation for multidimensional factors. When you use simple logistic regression versus complex black box, random forest, we compare different types of methods and different ways of explaining which one improves XAI, explainability, right? And so we had access to interesting data set. That's what allowed us to do this kind of testing. So, you'll hear the term AI explainability, but we need to test it empirically. What does it mean? What is AI explainability in context of care, right? So we actually ran experiments with clinicians explaining things in different ways and try to figure out what factors contribute to improving the explainability or decreasing explainability. So this was accepted recently in this IEEE ICHI, and one of our colleagues is going to be presenting. All right, now we'll continue. This is used in epidemiology, this types of approach, syndromic surveillance, and we've also done patient engagement work, where patient education work, where we've applied adaptive learning or personalization to provide different ways for patients to search, learn the information that is important to them, and use a personalized approach to do the education. And the personalized approach is really important because just having, I don't know how many of you know, that there is a brother to PubMed or a sister called Medline Plus. Medline Plus is consumer-oriented database for healthcare. So we use this approach on top of Medline Plus to help improve the education. And this is a version of the sort of the broad framework we use. That is, we developed something called a user model. As user interacts with Medline Plus, we try to learn their interest and then customize the presentation according to learning the user model. And then we tested which kinds of modeling improves interaction with Medline Plus, right? Personalized education, as you know, is a holy grail, right? So how do you build the personalization? How do you test it? What kind of behavior data? As you know, you love Netflix, right? Netflix uses personalization in a very deep way. So we were using personalization approaches and testing it for patient education. Right? Okay, and there are multiple papers, but that's one of those there. We also tested, of course, conversational bots, which is very important these days, as you know. We worked with aging population to test how they find information on a patient engagement side, because aging population don't want to use technology very much. They just want to talk. So if they talk, what are the behaviors? What is the conversational dialogue challenges in terms of understanding their information seeking patterns? So one of my students and I did a long-term project. It took like 4 years that we published this paper on understanding aging population versus young population as they interact with bots for engaging with the information search. And let me go quickly now. So this is our data warehouse, by the way. Well, I say our data warehouse. This is UNC data warehouse, EPIC. And EPIC has associated with data that secondary analysis system called CDW. And then we collect all of this data, and the idea is to get into this learning health system model, right? Where we take data, we do secondary analysis, we learn new intervention, and then we apply intervention translation-wise. Do you know who developed this model, by the way? Sorry to sound like a professor, but this is a very important learning health system. Well, people may argue, but this was developed by someone who was at UNC for a long time, but now he's at Michigan. Charles Friedman, Chuck Friedman. Yeah, he's become famous just developing this model, right? Learning health system, this cycle, right? Taking data from here, creating new models and intervention, launching them, testing them in the real world. This is the CTSA philosophy, right? 2 minutes, okay. All right, so we've done other work in the secondary analysis. One of the big problem is data integration, but you know that data integration doesn't happen because people are scared about sharing data. So we used, encryption methods to do matching and bring data together. We worked with EPR database and our CDW. This paper was also published in EMEA. You can find how to do basically privacy preserving data integration. And this is an example that I think Mateen is very probably familiar with. It comes from our pipeline. We created a notion of pipeline and workflow on data. So we can create models and we can export models to each other across healthcare systems. But it requires a lot of work on making the infrastructure itself intelligent in interesting ways. So this is combining SES data with clinical data coming from different sites, but then creating a pipeline. I miss this project very much because we had colleagues like Mateen and David from multiple sites. I hope it is going well. This is the final one I will show that I'm really excited about, where we're also bringing in LLM in a big way. I've applied LLM, of course, before. Language models. Language models, just like many other techniques, are relatively old, as you probably know. But doing it at scale in a big way is very important. What am I showing? This comes from also Chuck Friedman's work on computable biomedical knowledge, CBK. So going back to where we started, taking literature, finding evidence, creating models from them, and then going to computable code, and then creating libraries of code, all automatically. So here's an example of how we extract guidelines from evidence, convert the guidelines to structured format, and eventually to code. And using MetaLlama, because it's one of the largest and open LLM platforms, to do that work. Once we automate this, we can deploy it using things like InfoButton with EHR. So last thing I will show is we need to sustain all of this. And Martina, I know, is very interested in this long-term plan. So this is part of learning health system. We need to create a community of learners, not just create the loop from data to discovery, to knowledge, to deployment, to new data, but we need to create a community around it. And it's part of learning health system, this formation of learning community. And I have done this. I was lucky to have achieved this at UNC. We've launched 3 different degree programs under the CHIP program. And we have also done online learning in a big way. We were able to partner with United Health Foundation to do that. And as I mentioned, I've found it with students, and my colleagues have found it, just like it's happening here, spin-offs. Right? So this is a summary of everything we talked about, the pipeline with AI/ML, and going into that learning community is really important for long-term sustainability. Thank you very much for your patience.
Thank you so much, Javed. This is very inspirational. I think a lot of people will be looking at their research pipelines and then saying, which one of these are A to B, which one of them are B to C, and I certainly expect 2 papers a minute this afternoon.
Maybe we should start next week.
Monday.
And I don't know, just we are severely over time.
Oh, sorry.
And
There's
I
no
don't know.
questions in the chat.
Okay,
Yeah.
so if people are here, if you have any questions, please